Okay, welcome everybody. Thank you very, very much for joining us today. We're very excited to have you with us. Quickly, before we dive into the exciting program today, just please let me know that you can see the screen, that you can see the slides, and that you can also hear me okay. You can do that by just dropping a quick note in the chat saying hello and telling me where you're calling from. I think we've got people joining us from all over the globe today, which is very exciting indeed. So let me just go ahead and just check the chat, make sure you can hear me and that you can also post questions and everything in there later on. Okay, there we go. We have some coming through now. Very good, very good. All right, welcome to this webinar, and it's my great pleasure on behalf of SimCell to introduce you to Kyle Cichos. He is the Vice Chair of Research and Director of Translational Research at the Hughston Clinic, and he is, I think it's fair to say, a calScreener super user. He is doing really, really amazing things in the field of orthopedic infections with microcalorimetry, and it is really my pleasure to introduce him to you today to hear about the exciting things that he is doing and how he's pushing the boundaries of infection detection. So thank you very much, everyone, for joining. Thank you, Kyle. Over to you.
Thanks, Ben. I appreciate the introduction. And hopefully we can get this talk started here on improving accuracy and time to detection of bacteria for orthopedic infections, in particular focusing on the use of calorimetry and how we think that fits in. As far as disclosures, nothing other than that SimCell did support some of our work on developing this process early on. So first and foremost, why orthopedic tissues? The conventional cultures that are considered today the gold standard have a 10 to 20% false negative rate, and also take up to 5 days for aerobic cultures to grow, as well as 7 to 10 days or more even for most anaerobic cultures. With that, we also have an extremely high number of orthopedic infections globally. There's a large disease burden in that sense. While it's not officially known or documented extremely accurately around the globe, recent estimates say that we have 3 to 5 million cases per year. And the cost to a healthcare system for one infection case is upwards of $100,000 per case. And even more importantly than that, the cost of the patient is substantial, with a 5-year mortality rate upwards of 25%, depending on the type of infection that we're dealing with within the orthopedic space. So it's a substantial burden that we thought we could do something to improve and maybe see if we can help with the overall outcomes of these patients by improving time and accuracy. Current practice is during surgery, taking 3 to 5 deep tissue culture samples at the time of surgery. Detection is then done by division into different culturing conditions and cultivated. As I mentioned, aerobic usually maintained for a minimum of 5 days, anaerobic a minimum of 10 to 14 days. Then ID typically is done with some form of multi-top or other speciation identification technique. And then antibiotic susceptibility testing usually occurs after that and takes upwards of 24 hours, sometimes as long as 48 hours, at least at our institution. So why calorimetry? So we thought isothermal microcalorimetry would be a potentially interesting technique to investigate in this space. The reason behind that and the technical workflow for that is that calorimetry measures metabolic activity as heat flow. So the theory behind it is if bacteria are alive and metabolizing, growing or not growing, as long as they're metabolizing, they're producing heat, and we can detect that if the sensors are powerful enough to do so. So we figure that would have a nice potential improvement on time, given the increased sensitivity to this, where they don't actually have to be growing. A lot of cultures and culture conditions and bacteria don't always work out very well together. The bacteria don't like to grow. They're hard to culture, difficult to cultivate. Sometimes they're slow growers, take a long time, especially in the orthopedic space. We see that with several of our more prominent bacteria that cause infections, that they become slow growers. And while they may not be growing fast enough on culture plates, calorimetry has the potential to detect them the entire time while they're metabolizing in the process. The sensitivity is in the microwatt range, and you end up with a thermogram, as you can see on the bottom of this slide here, that sort of depicts the different stages of the bacterial growth process. So to date, we've done a significant number of orthopedic tissue samples using calorimetry. We've done more than 400 patients, more than 1,500 total tissue samples with all sorts of different patients within the orthopedic space, all the different subspecialties and really areas of the body related to orthopedics as well. With those samples, we've done a direct comparison to standard conventional tissue cultures, as well as in around 30 cases or so, we've done next generation sequencing as well, to just have a sort of baseline or pilot data looking at that. So our general process, lumped a lot of this in together. We've published a couple papers on this now, but I kind of thought it would be good to go over the process in general up front, as it was very similar process used for both. However, I also will put the caveat in there that during this process, we were sort of developing and optimizing it. So some things changed early on. It took us a little bit of time initially to hone in on a few specific details, such as run duration and things like that. So the data that we see includes the trial and error process for that. So there are some limitations in what we were doing early that may have impacted how effective the calorimetry was. But I think that doing it this way allowed us to really hone in on the overall process that we arrived at today. And so I'll kind of highlight the things that we settled in on as well as we go. But of the 3 to 5 tissue samples that are obtained intraoperatively, they were always sent to our hospital lab for standard culture processing first, as part of the traditional standard of care. They were homogenized mechanically and individually, so there was no pooling of samples. Each sample is then aliquoted equally for conventional cultures, isothermal, microcalorimetry, and then again, in the 30 or so cases, there was also an equal aliquot that went for NGS as well. The calorimetry and NGS aliquots were transported to our research lab, where they were then further processed immediately, and the culture ones were continually processed at the lab in the hospital as normal. So what did that look like for the conventional cultures? That involved blood chocolate and mechanochyography for aerobic incubation for 5 days, bioglycolate and Brucella bloodograph for anaerobic incubation for looking at anaerobic cultures. We also had gram-stained AFB and fungal cultures for every sample, and then species ID was from colonies using MALDI-TOF. For calorimetry, the homogenates were filtered through a 70 micron strainer and centrifuged, and then resuspended in 1x PBS. We used 40 microliter aliquots of these that we loaded into CalWell inserts with media. We tried to do some varying media throughout this process. As you can see below we used TSB, MH, BHI, YPD, Thio, SDV, and salt mannitol at various points throughout the process. TSB was used on every sample as kind of our baseline aerobic media, enrichment media. We chose TSB in comparison with MH because it seemed like it was the best for bacterial growth, and we wanted to test the limits and see if we could increase the speed and time while reducing the time to detection overall. So what we ended up settling on for the majority of the samples was 4 different media types. We used TSB, Soberads, bioglycolate, and salt mannitol to try to give us some distinction between different pathogens and different growth conditions. The total sample volume was 300 microliters for aerobic cultures and 400 microliters for our anaerobic cultures. And the run duration was a minimum of 24 hours in the calScreener. That was, again, an evolving process. Early on, we set it at basically 24 hours, and we were running pretty consistently right around that time. We learned that there were some that were slower growing and needed just a little bit longer that we were missing pretty clearly from that. And so we extended that run duration and eventually started running it for a minimum of 4 to 5 days to, again, further evaluate the optimal duration of the runtime. So a minimum of 24 hours. We've run it as long as, I believe, 26 or 7 days, just again to see if we would start having false positives or things like that. The speciation occurred by multi-top from cow well pellets as part of a pilot. And before that, it was then we used subcultures and then went to multi-top as well to confirm that what we were growing in the calorimetry was identical to what we were seeing in cultures, or if we were growing something else. So the general process, again, upon loading into the calScreener, there's 2 states, an equilibration state that's 10 minutes, and then another 20-minute kind of equilibration in the final location where the heat sensing is going to occur. The heat production after those 2 equilibration steps occurs is continuously measured for the duration of the run. And then after the completion of the run, baseline correction is applied. And then a positive reading for this study was used as 10 microwatts above the baseline. So to get into a little bit of the data for this and the results for this, one of the first things we did, we looked at the ability to use it for fracture-related infection cases. We separated the orthopedic work into groups based on essentially the criteria that we were going to use. So FRI criteria makes it a little easier to explain throughout the methods process, but the overall design of the studies were quite similar. For this one, we had 97 total patients, 68 FRI positive patients. This is sort of a slide showing all the different cases, whether they were monomicrobial, polymicrobial, culture negative, and calorimetry negative. Really, the main point of this slide, though, is to just kind of demonstrate the breadth of different pathogens we were able to detect. There's quite an extensive variability here. We detected 30 different unique pathogens in this case. And again, we had the majority of these cases were monomicrobial, but we also had 19 polymicrobial cases that were included in this portion of the project as well. What we found in the results of this, the comparison, sensitivity improved, accuracy improved in the overall cohort of patients, and time to detection in hours. In this case, for the calorimetry, the median time to detection was around 2 hours, compared to cultures was 51 hours. So we substantially improved that, of course. Accuracy was not quite statistically significant, but that was really just a power issue ultimately. We have since run an additional 30 or 40 fracture-related infection cases. We've rerun the analyses, and the metrics stay very similar, but the accuracy becomes statistically significant. So overall, we think it's a pretty dramatic improvement in time to detection, while also eventually became an improvement in accuracy as well that was statistically significant. And the second table here is another thing that we thought was quite interesting, and I wanted to really kind of emphasize a little bit of that. One of the biggest problems with the conventional cultures that we use today are the sort of patients that are on chronic antibiotics, or patients that get antibiotics before sampling for whatever reason that may be happening. Cultures don't do a great job with that. I think, as most of us that work in that space know, they don't have the best sensitivity in that area. We thought the process of calorimetry may improve that. We didn't know how much. When we looked at it, it was a little bit more dramatic than we expected initially. There was really no change in the median time to detection for these patients. As you can see, it was very similar, but the sensitivity improved quite dramatically with the calorimetry, and accuracy, again, I think again, with a little bit extra power, that became statistically significant. So in this paper itself, we weren't able to discuss sort of a true improvement in accuracy, but it was pretty clear that the calorimetry was much greater than the cultures for fracture-related infection cases, and substantially greater for patients on chronic antibiotics in any form. We don't have a definitive reason for that at this point. We have some theories. One of them that we feel maybe helping out with that is the process that we described previously in the methods, where we are spinning it down and resuspending it. It may end up being diluting any antibiotics that are still present in the culture sample, and that may allow us to sort of detect the bacteria and allow them to survive in the calorimetry, as compared to culture, where that really doesn't happen. They just get plated. If the antibiotics are still present in the sample to any extent, it may be inhibiting growth and limiting that capacity. That's just our sort of working theory at this point, but it is a pretty dramatic improvement nonetheless. And then our second project was looking at the use of calorimetry for periprosthetic joint infection. There's a couple different criteria that we compared it to as sort of the gold standard diagnostic criteria in this space. The traditional MSIS criteria that was published in 2014. We also then used the updated 2018 criteria that included inconclusive cases. It had 3 levels of diagnostic criteria for that. So we wanted to compare it to both. Again, similar to the previous slide for FRI, we had a substantial number of unique pathogens detected. In this case, there was 26 unique pathogens detected in the 64 periprosthetic joint infection patients that we had. And again, the breadth of sort of variability of these cases between sort of slow growers, really fast growers, more virulent, a little bit less virulent, all of those things is pretty well covered here. The majority of these ones, again, were monomicrobial infections. However, there were 20 polymicrobial and 6 that were ultimately culture negative and calorimetry negative as well, that were classified as PGI. Wanted to include this slide really just to demonstrate that sort of the unique curves that each pathogen has, and how those curves tend to stay pretty similar for the most part on different infection cases. So every time that we are detecting the individual bacteria, we can compare it, and on the curve we can see that within the same media condition, the curves are generally similar. I think you can tell that they're unique enough to the naked eye that in many cases you can even see it when you first are watching it in real time visually with your own eyes. You can see it as opposed to even having to have a computer to detect differences. So these were the specific pathogens that we had multiple monomicrobial infections of, and wanted to keep the overlay of the slides there to demonstrate the similarity within each case of the same pathogen. When you overlay that all in one slide, you can see that there's a great variability, and that for the most part, even visually to the naked eye, you can detect the difference between the different thermograms produced by each species that we are looking at here. Some of them are a little bit more similar. I would say the gram-positive pathogens tend to have curves that are a little bit more similar to each other and a little bit harder to distinguish. The gram-negatives tend to have pretty unique curves. Like, for example, Pseudomonas, you can see is dramatically higher in terms of the max heat flow produced compared to anything else. It's pretty unique in that sense. So again, I think to the naked eye, you can see most of those things, and this slide I think demonstrates that very well, that the species have very unique curves in general. So the results of this particular case and study that we did looking at PGI, the sensitivity improved as well as the accuracy, and the time to detection also significantly improved in this study. The accuracy went from 87% with conventional cultures to 93% for calorimetry. And the time to detection, again, 10 hours on the median in this case, compared to 51 hours for the cultures, which is pretty consistent with the published literature on how long it takes cultures. I've seen anywhere from, I think, 48 hours to 72 hours for averages published in the literature for conventional cultures. So we feel like our institution was sort of within the range of what's been published for averages across the board. Again, we also then compared it to the 2018 criteria. We had 146 total patients in this study as well. For this part of it, 6, however, were excluded because the results were inconclusive based on the criteria definitions. That said, of those 6 that were excluded, 4 of them had one of the samples positive by calorimetry, and if we would have substituted calorimetry into the criteria in place of cultures, they would have been positive by calorimetry, and it would have improved the accuracy results back up to the 90, I think it was 95% range for those cases. So we saw a dramatic improvement in accuracy as it is with calorimetry, but even with the inconclusive cases, had we been able to substitute it officially for cultures, it would have been even better results than what we saw here. This is a slide for all of the samples that we've done within the PGI space to date, sort of trying to demonstrate the time to detection and how many of the PGIs were identified at which time points for both calorimetry and for conventional cultures. For the calorimetry side of things, around 75% of all of the cases were identified by the 24 to 30 hour time window, compared to cultures where that number was around 10% of all of the cases were identified by that time point. So I think it's a pretty dramatic improvement and pretty significant that we can identify approximately 30% of cases within the first hour on the calorimetry, and that we continue to improve that detection to where by 12 hours, we've identified more than 50% of all of the infection cases that we studied on PGI. Again, for the same, similar to what we did for the FRI portion of the project, for PGI, we also wanted to look at patients who are on chronic antibiotics. These were not patients who were on spacers or anything like that at this point. We excluded those patients from the analysis or from this project based on our initial inclusion and exclusion criteria in our study design. We do have patients with chronic spacers and long-term IV antibiotic users that we have data on. We just don't have enough to really analyze and make sense of it yet. But for this case, these were patients who had implants in place and were on chronic IV antibiotics or oral antibiotics for more than a week at a time in most cases prior to sampling. We looked at that, and again, we saw similar results to the FRI case where we improved the accuracy. The time to detection was, again, pretty similar to the results of those not on antibiotics, but the sensitivity and accuracy increased substantially for these cases. And again, I think it's something that was a little bit unexpected, but pretty significant in the overall scheme of things, because for a long time we've known that that's an issue for cultures, and we know that that's one of the things that's potentially accounting for the limitations that we have in overall accuracy of cultures so far. So that said, there are some limitations to what we at least currently know about calorimetry and how we can use it for orthopedic infection detection and hopefully improvement in the ID portion of all of this. But one of the things that we still have to optimize is performance for anaerobes and develop a good protocol for that. We initially started with the thioglycolate media wells and thought that might be something that can give us a chance. We have detected anaerobes using this method before. We just don't have enough overall cases or instances of that yet to really hone in and say that we have a definitive protocol in that sense that we know works well. We know how we have a protocol that does work a little bit, but early on, and again, these patients were included as missed cases for calorimetry in the data that I showed before. But early on, we weren't even using thioglycolate. We were just using TSB at the initial steps of this process. And there were at least 2 or 3 instances of truly anaerobic pathogens causing the infection that we didn't detect, and we wouldn't have expected to with the setup that we had. And later on, once we developed the thioglycolate portion of it, and as part of our investigation, we only actually encountered, I believe it was 2 cases of anaerobic pathogens at that point. Excuse me. One of those we detected with the thioglycolate wells, and the other we did not, for various reasons, potentially related to the handling of the sample from the OR to the lab. But nonetheless, again, I think that's something just to note, that is an area that requires further development as we move forward, and optimization likely. Another potential limitation is the use of calorimetry in other sample types. We haven't experimented it in this data here with synovial fluid, urine samples, sputum, anything else that traditional cultures are used for. So those things I think are areas for expansion of calorimetry use, but again, would require further study and protocol development, process refining to some sense. I know there are some prior publications out there in those areas using other calorimeters, and they look promising to date. So I think it can be done, but it's something that we haven't done in our hands, greatly, to a great extent at this point. So can't comment too much on that from my side of it. Antibiotic susceptibility testing is another, I think, sort of next step, sort of process with calorimetry. And I think that is going to be vital to the overall time improvement, to being able to take the whole process from start to finish potentially in a calorimeter is something that would be intriguing and just needs a little further optimization. I know there have been multiple publications to date using calorimetry for antibiotic susceptibility testing. So it seems like it can be done most likely. And again, we haven't done that in our hands yet to attempt taking it from start to finish. So I think that's something that may require some work, but seems to be promising to this point. And the last thing really has 2 parts to it, which was speciation by the curves alone. I think one of the limitations that we see with the early portion of the process that we have been doing was that the need for sort of a reculture afterwards to truly speciate the pathogen. We could detect it, know that it was positive, know that the patients needed to be on some type of antibiotic definitively for a PGI case. We just couldn't, I think, 100% confidently delineate it just with the naked eye alone in every situation. And so I think further work needs to be done on that. It's underway already, at least from our angle of things, and it's very promising, including some, version of large language models and machine learning type of algorithms that can help with that. The data, like I said, to date is very promising, but it's something that needs to be sort of further improved upon as we go forward. I'm not sure that we can, at this point in time, do that. We did, as I mentioned, do a little pilot using MALDI directly from the liquid pellets, and that seemed to show some promising work as well as a potential stopgap until we can use curves. But again, it was a small number of samples, and polymicrobial samples in that case are challenging. MALDI's not able to do that from the same pellet itself, multiple different bacteria. It doesn't do well with that, and so that's generally where the separating out the individual colonies comes into play with traditional cultures. So that part needs a little bit further optimization as well. And then again, as I mentioned the second part of the speciation by curves is really polymicrobial infections. I think that eventually, if we have the right combination of media, which is what we were striving for through some of our experimentation with that, and further optimizing that, may allow us to be able to truly detect the different pathogens by curves alone in different media combinations. But that again needs some further optimization and study going forward before we can definitively say that speciation can be done confidently on the curves alone. But although, like I said, it is promising what we've seen so far. So I think the bottom line that what we saw is that the calorimetry has the potential to sort of lead a shift in how we actually detect PJI and FRI and other infection cases within the field of orthopedics, especially when it comes to tissue cultures. And the biggest component of that, as you can see from the timeline on the bottom, is really the detection stage of bacterial presence. If we can save multiple days on that through the use of calorimetry, it will really just expedite the whole process to IV and antibiotic testing. Even if we have to do sort of the current standard of reculture and all ID afterwards after the calorimetry, because of the process of calorimetry, because it doesn't destroy the bacteria, it doesn't really harm them. It basically is just incubating them and allowing them to grow. We see that the actual growth on plates happens, much faster than the 50-hour timeline average. So we're even in those cases, we're able to have colonies typically in the 12 to 14-hour range that we can pick and identify for using multi-top, and then you can go into the traditional antibiotic susceptibility testing with the lab. So we found that the roughly 2-day average improvement was maintained, even using that process. It was maintained for the most part compared to the conventional cultures in the long run. So as we optimize the other portions that I mentioned, I think that there's potential to continue to improve each aspect of the standard processes that are demonstrated across the bottom of the slide here. With that, I just want to thank Simcell for having me here, and would be welcome to any questions at this point.
Thank you very much indeed, Kyle. It's so inspiring to see how you're really kind of pushing this in what's a really challenging environment. Your clinical microbiology is it's nothing easy. There's nothing easy about it, and it's really, really cool to see what you're doing. So yeah, please jump in the Q&A or the chat with questions. We already have a lot of questions all ready to go, Kyle. So I think people are coming in at different parts of the presentations. This may go back a little bit to things you were saying earlier on. But for example, why did you select 10 microwatts above the baseline as the criteria for positive samples? And is the background noise with clinical samples, is this higher than you would get with media alone?
Yeah, I see that. Thanks, Tom, for the question. We selected 10 initially because the few prior publications before we started our work that used calorimetry, they were using a different calorimeter to my knowledge, but they used 10 as the baseline in their work, and we felt like we would at least start with that. We're currently actually going back and looking at that. It's probably the next study that I would imagine we will submit for publication is sort of what is the optimal cutoff for detecting the infection. We think based on what I've seen so far from the data, it's not the analysis isn't completely done yet, but I believe we can actually bring that down to somewhere in the 4 or 5 microwatt range above the baseline. The background noise is actually pretty low regardless. And so it really seems like even bringing it down to the 3 to somewhere in the 3 to 5 range is not going to cause any real false positive problems or anything like that. So seems like we can bring it down, but that's something we're currently working on, further investigating.
Great. Are there any other factors you saw affecting the baseline?
Not really, honestly. The baseline is unique to each run, and so that it's based on, I guess, a point that are picked at the start of the finish. But in general, everything has a plateau stage. Again, either early on, if there's very few bacteria in the sample initially, and definitely at the end, if you allow it to run long enough, it returns back to baseline. So didn't seem to be a huge impact on that. We did look into the use of whether human cells, eukaryotic cells that may be present in the tissues were impacting that and causing some levels of increase, and that's probably the best answer I can give as something that may cause the baseline to sort of be a little elevated in some compared to the others. But we didn't see a major impact on that. It generally stayed, as I just mentioned, below the 3 microwatt range at most.
Great. Thank you. So we have a question here comparing to the field of water microbiology, is saying that they just use enzymatic measurements to detect contamination. So why would that not work here? I said 15 minutes actually from sampling to get a result. Like, why would enzymatic approaches, assays, not work in this case?
Yeah, I'm not I guess super familiar with all the different enzymatic assays for this, but I do know that one of the goals for several things is to look at things that are produced at antigens. I know antigen testing is one that I guess is probably not terribly far off from that for different bacterial species, and that's been looked at before. It has some pretty good results on improving detection for orthopedic infections. But yeah, I think it's a good idea. It's something we could look into further in the future, and probably the field of orthopedics could look into. But I have not heard of that actually used in the clinical space very much at all, to be honest.
No, neither have I actually. It's a good question. I guess the samples can be quite messy, right? So there's lots of things going on, and one of the biggest arguments we have for the calScreener is that you're not adding anything in there. It's completely label free, so you're actually just detecting what's there. You don't need to add any reagents or enzymes or substrates or anything like that. So that'd be one thing. And I guess sensitivity may be an issue. We're extremely sensitive compared to many of the other methods we know about, and I don't know how sensitive the enzymatic approaches are, depending how you're detecting them, I guess. But okay, but really good point.
Agree, but
anything that makes you think differently is nice, isn't it?
Absolutely.
Do you have any experience with intracellular bacteria? I've got a question about whether it can be used detecting intracellular bacteria.
We haven't actually specifically studied that question. However, within orthopedics, we do know that there is a not insignificant sample, I guess the best way I could say it, a fair number of cases where the bacteria are intracellular, whether that be in bone or any of the other tissue cells locally that we deal with on a regular basis in orthopedic cases. So as far as that goes, it's a great question. We haven't specifically done those in lab studies, basically putting the bacteria inside eukaryotic cells and testing to see if we're able to detect that. My guess is, if I had to guess, it's probably going to have a hard time initially detecting them until the bacteria start to try to reproduce or grow. If they're just kind of slowly metabolizing within the cell and in that sort of almost like a dormant phase within the cell. It's probably going to cause very little heat production or changes to that, and may be difficult to detect. Like I said, you can see from the data, it's not perfect at this stage. So all of these, I think, are very, very good points to investigate a little further, and that would be something we could look into in the future.
Yeah, that's super interesting. I was just desperately trying to Slack with some of my colleagues there to see if we have any data on this. I don't think we do, but just to reiterate what you say there, Kyle, I mean, there's going to be no issue with the background of the eukaryotic cells. They're not, if the bacteria kind of metabolizing, you'll definitely see that the eukaryotic cells will not really interfere. But exactly as you say, I think it just depends what they're doing intracellularly. If they're kind of dormant, then there's nothing really to detect because we're detecting heat and metabolic activity. But if they start doing stuff, if they start moving or metabolizing or producing proteins or dividing, then we could see that for sure. Okay, how about using calorimetry to guide antibiotic choice? How are you thinking of using it? Can you use it already?
Yeah, I think as I mentioned for the antibiotic susceptibility testing, I think it can be done through the use of calorimetry. I think some of the goals are to investigate further whether we can go directly from the wells that we are using for detection and speciation and move that right into sort of a group of wells that would have the different antibiotics in them already that are pretty standard that we use for antibiotic susceptibility testing in the current methods today, and then use that to determine the susceptibilities to each one of them. It's very similar. Inhibition basically produces no growth at all on the thermogram, so you would know that again that the bacteria would be sensitive to that antibiotic, and if you get growth and heat production through the thermograms, then that antibiotic would not be effective against that particular pathogen or bacteria species. So it's very similar to the current methods, I guess, in that sense. But I think it's something that we are looking to explore further, the process from the detection wells to the susceptibility wells.
Great. We have a detail picked up here. I think you can tell the people have been using the instrument before. So what about the difference in the initial volume between aerobic of 300 microliters and anaerobic of 400 microliters? What was the rationale behind the different volumes?
Yeah, so great question. I guess the initial thought behind it for the anaerobic side of it in particular, 300 was sort of the standard. We wanted to make sure we had enough media in there to support growth and metabolism by the bacteria so that we could try to expedite the time to detection. For the anaerobic side of it, we really went with 400. We tried to essentially fill up the cal well insert basically to prevent or to limit the amount of air that would be in the well when we enclosed it in, and hopefully try to help create a little bit more of an anaerobic environment, or as much as possible, without the initial start of an anaerobic chamber. Again, that's another portion of the process that we plan to explore going forward, is whether we actually need to do those process those samples that are going to be for anaerobic testing in an anaerobic chamber to help make it a truly anaerobic environment. We're not sure yet, but I think it's something we're going to try to get to in the future when we optimize that portion of it. But the 400 was to limit the amount of air in the vial while the bacteria and everything are in the calScreener.
Cool. We have another question about background, and I think we answered it, but I'm not 100% sure, so I just want to come back to it. The background, is it caused from the sample that you put in or the media? Just really quickly, because I think we definitely touched on it, and I think you answered this, but
I guess the true answer is potentially both, but the main driver of it is probably going to be the sample itself, most likely, especially eukaryotic cells that may be present in the sample.
Cool. Great to see clinic results of IMC. Do you really think this will be a better method than molecular tests for identification of microbial species?
I mean, there's been some in the orthopedic space. There has been some challenges with many of the molecular tests to date. Like I said a little bit ago, I think honestly one of the more promising ones that we've seen is actually the antigen, the species specific antigen testing in orthopedics. Anyways, that seems to be one that has the results have been pretty strong. What we've seen in using PCR based methods, what we see with things like NGS methods, especially in orthopedics, is a huge variability in terms of accuracy, sensitivity, specificity, all of those things. That's been published in multiple studies within the literature. Most places do not do PCR based testing as a primary go-to within the field of orthopedics for tissue cultures. It would be a send out or a special request at most places. I know at our institution, it's really a last resort if we're concerned of infection and we just can't detect anything on cultures multiple times, then they'll try to use those methods to sort of last resort that process. I know in our hands and in our analysis, what we did with the NGS, I can tell you the NGS data is extremely poor. I didn't include it here because again, it's more of a pilot. But we saw of the, I think 30 cases that we had NGS used, there was, I believe 6 false positives detecting pathogens that in cases where there was no infection present, and those patients have been followed beyond a year. So at least there's no acute infection. If they come back in 5-10 years and there's a chronic one, then I guess that data would need to be amended. But it's a problem that we've seen. We've seen it in the literature and other places as well. We also had an issue with NGS where there were several cases of the remaining 20 mid-20s or whatever that we've run. There were several cases where we identified the exact same bacteria growing in cultures and in calorimetry, but the NGS didn't even detect that pathogen. It detected other less virulent pathogens, but not the ones that were detected. And we're talking pathogens like E. coli. We're talking things like Staph aureus that were just completely not detected on NGS. So I think there's some issues there. And then at our institution, I would say because of all of that and because of the current issues that they've had with it, there's really a lack of adoption of the molecular testing. So this was much easier to have a conversation with. Everybody was interested in what we were doing. They believed it because bacteria were growing, I think, was one of the biggest portions of it. People didn't really believe a lot of the molecular tests for all the reasons I mentioned. They knew that there was a lot of false positives. They knew there was actually false negatives, and also just misidentification of pathogens was also an issue. So those 3 things had a lack of trust in molecular tests. So I think it does have a chance to be adopted and better in the long run.
Nice. That's interesting. I had no idea that they were that bad, or how things like this can cause everyone to be skeptical. I mean, it's it's interesting.
Ours was a small pilot, like I said, which is why I didn't really include it in here. I think when you get to bigger numbers, it may kind of wash some of that out a little bit, but I think it's still going to be a problem nonetheless, because those issues have come up in recent publications, at least in the orthopedic field, using those methods.
Yeah. Have a follow-up on the intracellular bacteria question. Are you processing the sample in order to release intracellular bacteria?
Yes, potentially. When it's being homogenized, that in theory should release that intracellular bacteria. The only caveat from my standpoint in the comments on that is really, we're not looking at all of the tissues under the microscope immediately before to definitively say that they're intracellular. So yes, in theory, we should be releasing them. However, I can't say definitively that we are, because we haven't looked to, prior to homogenizing the tissue to confirm location of bacteria presence or anything like that at this point. But we should be, to answer your question, yes.
I have another practical question here about how are you visualizing the experimental results for AST, because obviously there's going to be lots of different samples, lots of different causative agents. It must give you a challenge for the visualization.
Yeah, I think for the conversations that we've had with current processes and things that are being done, I think the flow could generally be, based on an initial question of gram positive and gram negative, to at least get you kind of into a series of appropriate or more appropriate, I guess, antibiotics for those types of pathogens as a primary starting point. And then, kind of honing in on a series of the most common antibiotics that we would use in those spaces, and loading the wells with each of the wells with the appropriate amount of bacteria and running it through. I think it can be done. It is going to require some optimization, as I mentioned, just to see how that works best from the detection part of it into the AST, whether we can do it directly, whether we have to do other intermediary steps in between. Early on, it looks like it's possible, but again, part of it is the thermogram and the identification of the species. So being able to do that, and at minimum move them into the buckets of sort of gram positive versus gram negative, I think helps dramatically in setting up the next steps with AST based on that. But we can also then, if there are species that we know are generally more resistant to certain things, we can further subspecialize sort of panels for that, so that we can have vials and inserts that have all of that pre-prepared and ready, just to add the bacteria into. So you're sort of just selecting it as you go, after you know what the detection and ID results are. I think that's kind of the initial visualization of that, is to use it based on the ID and detection, and select your agents appropriately based off of that.
Great.
Good question though, and something we definitely need to, as I mentioned, sort of hone in on and investigate further.
Fantastic. We have another question here in the chat. So in your hands, what are the minimum number of bacteria that can be detected, and how does the state of the bacteria, so if it's a biofilm or persister or something, how does that affect the IMC profiles? I can also fill in a little bit here with some of our data.
Yeah, minimum starting bacteria, I guess challenging to know coming from tissue samples directly. We don't really, it's one of the biggest challenges with actually doing it with tissue samples compared to doing lab or lab derived studies with it, is you don't know the starting volume. And so that was, I think, one of the things that we were initially investigating, where we thought 24 hours may be sufficient. We learned that certain either slow growing bacteria or maybe just a small initial inoculum takes a little while to get to that, and sometimes 24 hours wasn't quite enough duration on the runtime early on. We definitely missed a couple. I can think of staph epi being one that's typically pretty slow growing, and I know we missed a couple of those that were likely present and would have been detected if we had extended the run a little bit longer. But again, we learned from that. So I can't comment specifically on the tissue side of it, exactly what the minimum number is for bacteria. I do know that, again, the more you get, the curve basically gets interrupted. It gets shifted so far to the left that it gets interrupted. And so you don't actually see the initial part of the curve. But can't comment on the minimum specifically in tissue samples. The state of the bacteria definitely affects the curve, gives a little bit of an alteration to the profile, especially biofilm compared to planktonic. So if you have a biofilm already formed, from what we've seen, it's formed a biofilm in the insert or on whatever sample, whatever you're putting into the insert. you will see a slightly different curve than if you see that on a planktonic. In this case, all of the bacteria would be resuspended anyway, so there should be limited variability, I would say, on these tissue samples with that.
Cool.
Specifically to what we're doing.
That's great. Yeah, it's extremely challenging there. We see in most of our experiments that limited detection is essentially one. We can detect one bacterial cell, but you have to wait. So it depends on how long you leave it and they grow for. But typically, we'll see signals much earlier than you would on a plate, for example, or in a broth or something like this. Exactly what you said. Exactly, for the state of the bacteria, you do see differences. But I think the biggest striking difference between a biofilm using IMC compared to other methods is that in a biofilm, you don't have to worry about the physiology of the culture affecting the readout like you do with colony forming units. If you have a biofilm, you can never really be sure that, you can be sure that one cell is not causing one colony. There's, however you process them, aggregates to a certain degree, but of course that's not an issue in the calwell, because everything is just growing there as it's happily doing, and you measure the heat from every metabolizing bacterial cell. So I think that's one of the most interesting differences in terms of physiology that we see, and that you might see compared to traditional methods, that the results are very different. And that's because on plates you really struggle to count properly what's going on, whereas we can do a good job, I think, in the calScreener.
In the lab, sort of the non-tissue specific work that we've done with the calorimeter, it seems the biofilm tends to be a little bit less on the overall max heat flow. It doesn't quite get to the same max, but it does seem to persist longer and doesn't really return to baseline quite the same way with the planktonic. But again, that's sort of anecdotal from some of the other work that we've done, and not specific to the tissue side of it, where again we're doing the processing as mentioned from the tissue samples. In theory, they're really, everything is essentially resuspended in planktonic at that point.
Interesting. Well, I'm just looking at the time, Carl. I wasn't necessarily expecting us to go this long with questions, but everyone has been extremely engaged. And yeah, thank you so much, everyone, for being here, for asking your questions, for listening in. And thank you to you, Kyle, for such a fantastic overview of what you've been doing, and for engaging so much with all the questions. It's been an absolute pleasure. And with that, I say thank you and goodbye. And we'd love to hear from you all via email if you have any follow-up questions or anything like that. So thank you very much. Bye-bye now.